we're looking specifically, the European Space Agency has a great program right now that they have Sentinel-1 imagery, and it's open access. So that means you can actually go make an account and look at some of these amazing images of our oceans, of our land. So I created a data set using these images, and this is the pre-processing steps that I had to do. So specifically choosing a polarization. This is particularly important because certain polarizations in satellites, when you're collecting data, are more sensitive to certain features on the ocean or Earth's surface. And then, of course, you have to calibrate your data. We need to make sure that the way we're looking at the data not only is important for the model to learn, but also for us to take things and learn from that. And then, of course, normalization. And we chose our patch size of 512 by 512 pixels. And then model training. So, of course, that's the next step once we have this pre-processed. So through our investigation, we specifically looked at a segmentation architecture. It's known as the UNet model, and this model has been extensively used in the realm of biomedical imagery. It was actually made for the purpose because this type of model is great for data that has a very noisy features to it. So I just took a little screenshot of some of the image patches that I've been processing through my training. So you're able to see kind of, okay, what is my model looking at and kind of understand what it's working with. So as you see, specifically, not only is it looking at images such as an island or interactions with the sea, but also just patches that are open ocean or could be other features. Also, specifically in SAR images, as I previously spoke about, there's inherent speckle noise. So the really biggest thing we found through our findings is our models are great at looking at the islands and extracting those features near those islands because it kind of uses it as a indicator that that's a region it needs to look. But our biggest problem area is looking at the regions that are away from islands or maybe might look like speckle noise. So this is kind of just an overview of results because when you're looking at segmentation architectures, you need to understand your evaluation metrics. I'm still new to the field of deep learning, so I'm still kind of learning all the segmentation metrics, but these were two that were very prominent in assessing the segmentation architecture specifically. I provided just an image so you can kind of understand visually what these mean. And as I'll show in the next couple, the results, they're specifically looking at IOU, which is just looking at how much is our result actually matching what we're training the model. And then a dice coefficient is just looking at the area of overlap over the total area that we're looking at. So these are just two of my results that I have taken out of the high resolution model. So this model was trained on the entire synthetic aperture radar imagery. What we did is we took the entire image and patched it into 512 by 512 patches, and we trained it and then, of course, tested it on different images. So these images are actually not seen by the model. So you need to make sure when you're building these deep learning networks, when you train the model, you also want to test them, but not on something that the model has seen, or else you might get great results, but it's not going to generalize to maybe other images or other data sets really well. And then, of course, speckle noise. This is something I talked about previously, but speckle noise is something that I found to be an issue within my segmentation because when you're looking at some of the open ocean, it really can kind of create these different features that inherently the model wants to pick up as a wake, but they actually can just sometimes be something that we want to have as a complete black mask. So in this case, the predicted mask was actually supposed to be entirely black, but we actually had some artifacts that were picked up. So after we have trained our UNet high resolution model, we realized that it works great, but not in all areas, right? There's always room for improvement. So we decided going forward, we would implement a two-pass system. So essentially a two-pass system just means that when you're training your model, I'm still utilizing the high resolution model, but then I'm also training a low resolution model that will also help with getting those course predictions. And then what we essentially do is we combine those predictions together. So it kind of helps to average out and to resolve maybe those features that you see in the SAR image that aren't just near the islands and in the large scale. So it's really capturing those. So this was just showing one of the calibrated SAR images and how we downsampled it. So the difference between this system and the system that we did in the first pass is the second pass of the low resolution is actually trained on the entire SAR image. And this was something that my advisor really helped me understand, because when you're training such large imagery, there's so many features and so much to capture within them, that we're working on actually training the first pass on the low resolution to try to train the model to capture and see the really breadth of our image, especially the complexity within it. So this is just kind of our next step approaches of where we're going. So we're looking at a 2-pass system. We're first trying to look at maybe creating a false color image, which is combining our high resolution channels and, like I said, the upsampled lower resolution channels still within the unit to make segmentation predictions. And then secondly, doing the same thing, but instead of utilizing a false color image, at the end, we're doing segmentation predictions and multiplying that by the low resolution feature maps. And at the end of this, I just had a little summary of kind of where we're going from here. So we're looking at, of course, getting better results with the boundary delineation. I eventually want to look at saliency maps, because in the world of deep learning, results are great, but I really am interested in explainable AI and trying to understand behind the scenes of the models. And then lastly, radon transforms. And I know I don't want to go too much over time, but just that's the next step. And we just want to use radon transforms to kind of, again, like look at those linear features, and because UNet models can benefit from these additional input channels from those. So thank you so much. Thank you.
Questions for Madeleine?
You said that it's so it's a segmentation problem, and I missed how you labeled the
label,
the pixels in that.
So that was actually
Okay, I wasn't sure if
manual annotation. So that was by hand. So I worked with not my IDSC advisor, but my primary advisor. He's working in the realm of remote sensing and ocean science. So he helped me with the manual annotations with those. But those are very time consuming. So that's also another restriction that I didn't discuss, but that's kind of the motivator of why we're doing this 2-pass system, because I told my IDSC advisor, Dr. Chapman, it takes so much time. I really want to kind of refine my results. And so that's why we're looking at the smaller, so doing annotations for the whole image at a very small resolution and then feeding that into the model so it can help save with that time, because that's a great question. I hope I answered that. Okay.
Similar question. So you said that you trained the model. And you also mentioned that it works well for some things and works. So how many, you said you used all the data.
Okay.
So what does this mean? So, and can you elaborate a little bit of where it works and where it breaks, and any ideas of why?
Yeah, definitely. Yeah. So I think in terms of the data itself, so like I said, there's Sentinel-1 data, so it's open access. So I've collected about 500 images that I actually have, and then for this data set that I utilized, we utilized about 20,000 image patches. So those 512 by 512 image patches for the high resolution model. And then, so we utilize those for the training. And so what we found when we're only using those 512 patches from those such large images is what you're essentially doing is you're taking these small patches, but you're kind of losing that spatial context, right? Because when you're training that model, what you're doing is you're kind of losing that overlap is why we're going into the 2-pass model, because we really want to capture those features that, the island wake, as you saw in some of the images, it's not just in 512 by 512, it kind of extends throughout. So what we're doing is we're trying to, our next stage is training that model using that mindset on that. Did that answer your question or?
Yes.
Okay.
I have a question. Shouldn't the UNet be able to automatically sort of reduce the patches, do the multi-resolution passes on its own? Is this something that, or do you have to coarsen by hand first and then present it the other data sets?
So I think if I understand your question correctly, you're just saying Yes, essentially the UNet does do downsampling throughout it, through its processes. But what we're actually doing is we're talking about the input. Maybe I should have specified that. So the input imagery. So when we're processing this, essentially what we do is we take our 512 by 512, we patch that from the big images and we run the model and we say, okay, that's doing well. But what we do in the second pass is we take those full scale images and just run a code to downsample it and then run it through the image, run it through UNet, and it still does the downsampling and does the processing. So I think your question was maybe focusing on the architecture when we're really, all we're really doing to affect the size is at the beginning for the input. Mhm.
Can you remind everybody just the size? Because I think that's maybe another constraint in this problem. It's just the size of these SAR images.
Yeah, exactly.
And, can you kind of remind everybody, how large is a SAR image?
Right.
And then, also, what kind of data set do you have in terms of how many SAR big images and over how many scenes and things like that?
Yeah, oh, definitely. That's a great question. So in terms of the synthetic aperture radar data set we have, so first of all, the size. So they range about, on average with the satellite I'm working with, they're about 16,000 by 22,000 pixels. So when you really look at that, even on your computer that you probably have, it's very hard to see the entire thing, especially the high resolution details. And then also when you download the Sentinel-1 data from ESA, it actually is in about like terabytes. Like you have to kind of, for me as a researcher, it was really a big step in trying to learn the data, because you open it up and you're like, calibration files, measurement files, how do I want to calibrate it? And so, just going through that stage is a process that I know sometimes I think through my research and realize the time constraints. And that was kind of a big one at the beginning, saying, okay, how do we take this data and just streamline it? And I think through this fellowship, I got really good at getting faster with my processing. So that was good. But yeah, the synthetic aperture radar data is quite large. So in terms of how much I have on my computer right now, you can only hold so much, but I have about 500 images, full resolution images that I have. And of course, Sentinel-1's open access. So I can go next week and download a whole bunch more. But yeah, that's a big restraint on our problem, because we're trying to say, how do we streamline this while also, not being so resource constraining. But yeah, so I hope that answered it.
Yeah.
Okay.
Thank you.
Awesome. Thank you.
Thank you very much.
One more question. Oh, sorry.
One more question.
We'll have a time.
The end.
Yeah.
Yeah. Okay. Perfect.
Yeah.
Thank you so much. Well, thank you.
Our next speaker is Anna Golikova, who is majoring in biology and is graduating this semester.
Hi everyone, my name is Anna, and as I've been introduced, I'm a senior major in biology from biology department, and my project is about identification of bacteria viruses into rebuilding corals, Pteridopterus stigosa and Manterstea cavernosa. And my research supervisor is Cynthia Silveira, Dr. Cynthia Silveira from the biology department, and Dr. Ravi Vadapalli from the IDSC. So as you might remember, from the November meeting that they had, I'm going to do a little bit of an introduction and overview of my project. So we know that corals exist as coral holobiont, and it includes the coral animal itself and also its associated organisms such as viruses, bacteria, archaea, fungi, different protists, and the most popular are from the family Symbiodinaceae. And not a lot is known about viruses and how they interact organisms that live with corals and the coral. And we know in other systems that viruses can be involved in the pathogenicity and emergence of the disease, such as in a case of the cholera disease, when the actual toxin encoded inside the virus, which we call bacteriophage, that infects bacteria, and then it results in the disease for us humans. And we're thinking that the similar mechanism can happen in other systems such as coral reefs, and marine bacteriophages can in fact, coral bacteria that live with corals express their genes, and some of these genes can be responsible for the alternation of bacterial phenotype, which might make this bacterial pathogenic and affect corals. And in this project, I've been working with two important rebuilding corals here in Florida and also throughout the Caribbean, Tildiploria strigosa and Matrostea cavernosa. And the aim of my project was to look at this marine viruses that infect bacteria that live with corals. So my project had two parts, the part that I did in the lab and then the bioinformatic part. So as a result of my wet lab work, I was able to isolate viruses from concentrated from seawater, and I did it through plaque assays. When you put your bacteria that you isolated from coral tissues and mucus, you put them in a close proximity with viruses from seawater, and you're looking for infection. And the infection can be observed at this tiny transparent spot that you can probably see here, and we call it a plaque. And it originated from a single infection. So using this approach, I was able to find phages and isolate them for 3 bacterial hosts that turn out to be of the Vibrio species identified by the 16S gene sequencing. And Vibrio harvia had 6 potential phages, Vibrio acrolyticus 1 potential phage, and Vibrio that was not identified at a species level had 3 putative phages. Then I extracted the DNA from the viruses in their host, and I sent it for sequencing. And here is where the bioinformatic part came in. So this is a pipeline that I used for my methods. The data that I got from the sequences were the raw reads. So I used different softwares and I worked in the server to get the results that I have. And first step was to do a quality control of your raw reads, and I used BBDAC for that. And that helped me get rid of the poorly sequenced reads. And then I assembled this quality control reads in the bigger pieces that we call contigs using spades. And then I was trying to look for viruses within bacteria, which are the prophages, and the viruses within the isolates that I got. And I used Vibrant for that. And then I combined the data that I have, and I looked for how similar these viruses are. And I used Verathon for that. That gave me, that clustered my viral contig into viral populations based on the gene prediction that uses protocol to do that. And I got this viral population and I ran them through Feroca that gave me the different gene annotations. And I was interested in virulence factors and auxiliary metabolic genes that are encoded in phages, but are might be expressed in the host. And for bacterial genes, I also did the binning and the replication of them to obtain my bean contigs, and they turn out to be of 100% completeness and less than 1% contamination. And I used the resources of the Bacterial and Viral Bioinformatic Resource Center to do gene annotations. And here's some of the results that I obtained. So let's start with the bacterial genomes. I had only 3 bacteria isolates, and the most important and interesting was that I was able to annotate their individual genes, and the majority of annotations or protein coding sequences were identified within the F3, which is the first column in this first table. And it also had the longest genome, so it kind of expected. But the majority, there were a lot of hypothetical proteins that didn't have any annotations for all 3 bacteria. And I also looked at the specialty genes using these annotations, and some of them were similar to the other genes that might be responsible for encoding antimicrobial resistance or for products that might be gene targets or drug targets, or those that might be involved in a virulence. And for the proteins, for the genes that I had my functional annotation of the proteins, I looked at their distribution, and it was a little bit different for 3 bacteria, but the I saw that the majority belonged to metabolism, energy, and protein processing, and also membrane transport among this other subsystems. So those were the most prevalent in these 3 bacteria isolate that I had. And those are the genome, assembled genomes that I constructed. And from the left one is a F3 that we saw. The middle one is a Vibrio that wasn't identified at a species level, and the right one is a Vibrio choleolyticus. And here we can see the most outer ring, the blue one. It's just shows my context put all together in a circular genome. The green is protein coding sequences on the forward strand, pink on the reverse strand. Then turquoise shows some RNA genes that had annotations, and then there are some other annotations, as I said, that had homology to antimicrobial resistance genes here in red, to virulence factors in yellow, then to transporters in this blue, to possible drug targets in gray. And this is also the GC content of my genomes, and the most inner ring is a GCSQ, and those are the standard parameters for of genomes. And then I also wanted to confirm the taxonomical classification of my hosts that we got using the 16S gene sequencing, and it turned out to be that Vibrio, my F3 bacteria isolate was most similar to the Vibrio harrii. It was most related to it. For B11, it was indeed Vibrio choleolyticus, as we can see here. And for G4, they didn't have identification on the species level. Here we can see that it's most closely related to the Vibrio choleolyticus isolates that were available in a database, but it still forms its own branch. So, and then I wanted to look at the viruses that were identified. And as I said, the VRFON allowed them to cluster in 11 viral populations based on their similarities. And here's the distribution of this context for each bacterial isolate that I had. So on the left, the F3 had 4 prophages identified within the bacteria, and also 2 possible lytic viruses. The G4 had 3 prophages and 3 lytic viruses, and B11 had 3 prophages as well, and 1 lytic phage. And we can see that the virus that belongs to the viral population 2, it appeared in all 3 bacteria isolates, and also the lytic phage that's belong to the viral population 11. It also appeared among 3 of them, and this is what I'm going to be investigating further, and I'll mention that later. And as for putative taxonomy of the identified phages, I compared them to the GGI database that had environmental samples that different research shared, and both prophages and my lytic viruses were similar to double-stranded DNA tail phages. Some of them similar to single-stranded DNA phages, and some of them were filamentous phages in the Inaviridae family. And the viral population 11 contig was similar to the Microviridae family, which has a small single-stranded DNA phages. So I also did the gene annotation for this context within the viral populations, and here the most interesting results were that when I looked at the virulence factors, that using the tool, the PROCA tool that I used, didn't find any homologous to the virulence factors available from its database, and it found a little bit of AMGs, and a lot of genes were still of unknown function, and I didn't have any annotations for them, unfortunately. And let's look at the AMGs that I found. So for contexts that belong to viral population 9 and viral population 8, the one there was this gene, auxiliary metabolic gene which encoded for STB sensor of the two protein system, STB-STA. And we know that in other organisms, for example, in the Vibrio alginolyticus, it's been known to affect hemolysis and also virulence. And in other known marine pathogen of Photobacterium, it's been also known to play a role in virulence. Those contexts had the homology to this gene, and I'm gonna look closely to investigate how they can affect the relationship with their bacterial host. And there was also another AMG within the context that belonged to the viral population 8, and it encoded for a zolt-like toxin. And this is a known toxin that that's been identified in a Vibrio cholera, and it's been also known. It's been also found in different Vibrio species, both pathogenic or not. So I'm also gonna look closer on this specific gene that is within my viruses to investigate it further. And for future directions within my project, as I said, apart from the genes that I'm going to be looking more closely, I also want to do transmission electron microscopy on the lytic viruses that I have, and it will help confirm their taxonomy, because if they belong to tailphages, you'll be able to see the tail of the virus. And I also wanted to do a strain analysis of this isolated viruses to see for those that we saw infecting different bacterial hosts, to see if they're similar, if they're of the same strain of the same virus, or they actually belong to different strains. And with my information, I'll be able to look at the abundance of these viruses and bacteria that I identified in metagenomes of coral reefs here, isolated here in Florida and in the Caribbean, by using the metagenomes that we have in our lab, and also from the publicly available databases. And the overall, my study contributes to better understanding of the genomic potential of marine bacteriophages to be involved in the emergence of the coral pathogens through the alternation of relationships between viruses and bacteria that live with corals. And with that, I would like to thank the members of Silveria Lab that were really helpful throughout the whole project, my research supervisor, Dr. Cynthia Silveira, and my IDSC mentor, Dr. Ravi Vadapalli, and also the summer research scholarship from College of Arts and Sciences Beyond the Wood that funded part of my work. And with that, I can take the questions.
We have time for a quick question.
I have a question. A very interesting presentation. All right. Yeah, thank you. Very interesting presentation. I was wondering, you mentioned about different viral populations, and I was wondering, could you tell us just a little bit about were these viral populations collected from different samples from different corals, or were they from a similar geographic area? Or maybe you could just tell us a little bit about, how are these viral populations different?
Right. Yeah. So I looked at the viruses that were annotated within different bacteria, and also within the viruses that I isolated. And I looked at all of them. I combined all of them and wanted to see if they're similar or not. And then I used this program, Virathon, that basically it separated my context that I had. It separated into different groups based on their similarity. And to do that, it uses the gene prediction. It uses another program that's called Protocol that does the gene prediction. So it separated my data that I had into different groups.
Okay.
Yeah.
All right. Thank you, Anna. For the sake of time. Oh, one more.
That's okay.
That's okay. You have.
That's a lot of work. We're taking class at the same time, right? So I'm a little bit kind of clarification. But so you had 3 isolates for bacteria. How did you come up with this? And it appears that I didn't get.
Right. Yeah, I didn't cover that part.
Yes, how did you get to these three? Because I think,
Right.
you know, how do you get to these three isolates, the bacteria, which is individual bacteria or it was a mix of bacteria?
No, they were individual. So I actually started the project by, I had 83 bacteria isolates, but in our lab, we developed a protocol for screening of these bacteria to kind of speed up the process and see which can be infected by marine viruses. And we used the spectrophotometer basically using this approach. So that was the first part of my project that was, I didn't talk about it at all, but using this approach, I was able to first identify from 83, I was able to identify 20 putative bacteria that were possibly could be infected by the marine viruses from seawater. And then I did this manual testing that I talked about, plaque assays, and only 3 of the isolates, and you do it separately for each isolate, only 3 of them confirmed infection, consistent reinfection. So that's how I ended up with just 3.
Okay, that makes sense.
Yep.
Okay.
Thank you.
Thanks. Our third speaker is Christian Ramsoomair, who's an MD PhD student.
Is it okay to have to have the chat on the side or?
I'm trying to sort of close the chat.
Perfect. Okay, great. Okay. Hello, everybody. Good afternoon. My name is Richter Library, and I'm a third-year MD PhD student. And for this project, I was mentored by Dr. Shah, as well as Dr. Ravi Vedapalli in IDSC. So for this project, my main goal was to look at transposable elements in glioblastoma and understand whether they could be a novel target of therapies against this cancer. So for those that are familiar, glioblastoma is a grade 4 type of brain tumor that has a very dismal prognosis. Most patients survive less than 1 year after the date of diagnosis. So as a brief primer, whenever we think about like a solid tumor, all the different cells within within a tumor are normally different. And the way in which like the genome, the epigenome changes, like they're different. So within the past decade or so, there's been a model that has been proposed called the cancer stem cell model, where there was a small yet vital population of tumor cells known as cancer stem cells that are necessary for the self-renewal of the tumor, as well as the propagation of the tumor. So because of this, oncologists and scientists have been trying to figure out how do we target therapies against cells in this stem-like niche versus the other tumor cells, which are necessarily vital for the propagation of the tumor. So, you know, every cancer is different, like between brain, stomach, breast cancer, but there's been a lot of work trying to figure out what actually makes these cancer stem cells unique from the other type of tumor cells within the tumor. So they found that these cells are in fact unique in terms of the transcription factors within these cells, the enhancers within these cells, and the epigenome in general is very different within these stem cells. One understudied factor within what makes these cancer stem cells unique is the role of these transposable elements. So for the purposes of my talk today, I'll be focusing on transposable elements known as HERVs, human endogenous retroviruses. So these type of transposable elements are proposed to be oncogenic due to 3 different mechanisms. The first mechanism is this first one on top here, where these genomic elements are able to insert themselves within tumor suppressor genes, and this causes these genes to be silenced. The second way is that these retroviruses are able to insert themselves prior to an oncogene, and this leads to increased production of the oncogene in tumor formation. And the last way is that these endogenous retroviruses, there is some evidence to suggest that they create proteins that are known to interact with oncogenic pathways and thus also cause stem-like features within tumors. So normally within our bodies, HERVs or endogenous retroviruses are regulated and silenced. So some of these silencing mechanisms are histone methylation, DNA methylation, RNA methylation, and various kinds of silencing RNAs. However, when cancer occurs, regulation is known to decrease, and these transposable elements and endogenous retroviruses are known to become activated in the cancerous state. So the goals of my project was to just map what transposable elements, as well as what location of these transposable elements differ between cancerous tissue of the brain and normal brain tissue. Also, I wanted to look at what are the nearby coding genes around these loci of these activated transposable elements. And lastly, I wanted to look at publicly available single-cell RNA sequencing, which would tell us are these activated transposable elements more active in these stem cell niches versus the tumor cells that we know are not stem-like. So part of the reason why this area of study has been so under studied to date is because normally in RNA-seq data that is publicly available, transposable elements are not counted, and they're not they're not annotated. However, in the past 5 or so years, there's been an increased push to develop packages that are able to identify as well as quantify the amount of these transposable element activation within these RNA-seq data sets. So what I did was I took data sets available to my PI, where we have 71 samples from GBM patients here at UM, as well as we took RNA-seq samples from 34 patients from the NIH, where we had frontal regions of the brain, as well as 11 samples from the NIH where we had temporal samples from healthy individuals. So this was our general pipeline. So first, we aimed to do some QC on the sequence data. Then we aimed to do some normalization, as well as we used the package that I mentioned earlier to be able to identify as well as to quantify counts of these transposable elements. And then we did PCA analysis to filter out any of the outliers. And finally, we did a full change analysis between the elements in GBM versus elements in normal brain. And then we did a Welch modified t-test to compare the two. So here I'm just showing you a PCA plot of all the samples that we had. So in green we have the normal frontal brain samples, and in red we have the glioblastoma cancerous patient samples. And and on this side here, we have the pre-filtered, and on this side here, we have the post-filtered, where we filtered out some of the outliers here on the bottom. And then we made a volcano plot, where we have in blue here are the down-regulated endogenous retroviruses that we see in GBM versus normal brain, and in the red, we have the upregulated endogenous retroviruses, which we see in GBM. So just for context here, each of these dots represents an endogenous retrovirus, and the ones in blue are the downregulated, and the ones in red are the upregulated. So again, we did the same thing where we compared glioblastoma samples to the normal temporal brain. So again, we did filtering for the outliers, and then we also made another volcano plot where we were able to see the downregulated and the upregulated. So so I made a table about the top downregulated HERVs in GBM, as well as the top upregulated HERVs in GBM. And for the rest of my talk, I'll be talking about two of these HERVs in particular that we find of interest in the upregulated samples. So I just want to focus on LTR 5HS, as well as this MUR34B for the rest of my talk. So interestingly enough, LTR 5HS is a part of DNA which is known to control the activation of this type of endogenous retrovirus known as HML2. And my PI, Ashish Shah, has actually published on this retrovirus within the past year, where he's shown other proteins being made from this virus seem to be contributing to the stem cell niche within glioblastoma. So what LTR5HS is, it's a transcription binding site where we basically have our transcription factors that are known to bind to this spot, and it leads to increased activation of the downstream endogenous retrovirus. As OCT4 was found to be one of these transcription factors that is known to bind to this spot, and OCT4 it's known to be a transcription factor associated with neural stem cells. So things that LTR5HS, there are many different loci within our genome where the sequence is found. So what we did was that we wanted to see how much activated LTR5HS is there in GBM tissue versus normal brain tissue. And what we saw is that we see much more activation of these loci in GBM versus normal brain. And this is a figure from the paper that Ashish published last year, where he targeted a consensus sequence that is known to occur in all the HML2 loci within our genomes. And what he found was that when he targeted this, our DCAS9, which is a type of protein machinery known to suppress expression, he saw that in mice that have GBMs, that the survival was longer when we suppressed HML2 expression versus mice that were wild type. In this figure in here. So for my studies here, I have found that the most upregulated LTR5HS loci seems to be on chromosome 1 at this location here. So perhaps targeting this location in particular would lead to a more efficacious result than the result that we saw on the previous page for the Kaplan-Meier curve there. And the other curve that we saw that was quite plentiful in the upregulated data set is MER34B. So there isn't a whole bunch about this ERV in the literature, but I did find that it's also known to be upregulated in osteosarcoma. So perhaps also this ERV has some kind of oncogenic, almost stem-like factor to it, which of course is a very interesting research question to be studied somewhat down the road. So in conclusion, these transposable elements or non-spectral viruses are known to be differentially expressed between normal and GBM samples, and select ERVs appear to be correlated with the stem-like nature or aggressiveness of these tumors. And in particular, LTR5HS of the HML2 HERV-K subtype definitely seems to be an interesting one to target. Also, further analysis needed for me to see what are the overlapping genes around the loci that I identified, and all those genes also are contributing to stem-like or aggressive features as well. And finally, in the lab, in conjunction with the collaborators at NIH, we're developing therapies which are able to suppress specific loci within tumor cells. So perhaps in the coming months, we'll try to localize therapies to suppress this loci here and see if we're able to have increased survival in first mice with this mutation. So thank you so yes, thanks so much to the IDSC and for your attention today.
Questions for Christian?
Yes. So this is a sample.
So basically, so everybody understands. So basically, you use the GBM patient samples that are exclusively GBM from the collection that we have here at UM, and then the controls were from NIH.
Correct. Exactly. Yeah, because things that like to get RNA-seq samples of the brain is very rare. Precious. Yeah. Exactly. So things that like for other cancers, kind of like breast, colon cancer, you can always get a control, but things that for the brain, it's quite novel to be able to have a control sample. Okay.
There's a lot of brain. There's actually a very good brain bank here at UM.
Yes.
So,
yes.
Another question. So when you said about the regions that you got from NIH, are they the same regions that you had the patients? Because as you know, and probably most of the people here know, each region of the brain shows a different, characteristics, definitely different gene expression and so forth. So did you try to match that?
For sure. Yeah. So things that GBMs are known to occur primarily in the frontal cortex as well as the temporal cortex. So that's why we chose to use those two regions to compare as our controls. Yeah.
Okay. All right. Thank you, Christian. Last but not least, Tianhao Liu, who's a PhD student in biostats, will share his finding with us.
See my screen? Oh yeah, okay. Okay. Hello, I'm Tianhao Liu. I'm a third-year PhD student in biostatistics, and my mentor is Daniel Diaz, and IDSC mentor is Dr. Kamo. And today my topic is the data reduction using spectral analysis. So firstly, let's talk about what is the data reduction. The data reduction is reducing the volume of our raw data to make it more manageable and easier to process, while preserving its integrity and usefulness. It aims to facilitate analysis and interpretation by focusing on essential data segments. For example, in a data set of thousands of images, data reduction allows to analyze the representative samples without needing to review every single images. So we will see this later by example more clearly. So before I jump to my main algorithm, I'll talk a little bit about the background knowledge. Firstly, is the spectral clustering. Spectral clustering is a powerful technique in graph theory used for grouping similar data points into clusters. It utilizes the spectrum of the Laplacian metric, where the elements are eigenvectors and the key elements are eigenvectors. The process generally goes like the following two steps. The first one is to identify the eigenvectors corresponding to the smallest eigenvalues. And in my terminology, I would call it the PDS components. And in the second step is use these eigenvectors as features to drive the clustering algorithm. Usually it's use the k-means or whatever other clustering algorithm suitable where you lack can be substituted here. And the second component of important component of my algorithm is the PRIM, patient rule induction method. PRIM is a data mining technique used to find subspace of a data set where a target variable is optimized. Often for identifying regions with high densities of cases of interest. Here, we actually use this for density detection. And in some contexts, we usually call it mole hunting or bump hunting. And the algorithm has two steps. I'll introduce the most important steps here and left others. And it's called the peeling step. It iteratively peels off box of the data space where the target variables is not really high or low values. And here is definitely a high values of density. Here it's showing the this picture clearly that peeling and peeling, and in the end you end up to a relatively small region with a high density data points. Yeah. And here comes my algorithm for the data reduction. I call it a conditional version of PRIM, the conditional patient rule induction method. We call it C-PRIM. The algorithm is basically goes through like this. Firstly, using some cluster detection technique. In our context, is just a spectral clustering technique to find the clusters, like K clusters. And for each cluster, we do the principal component analysis for within a specific cluster, and then select the K leading components with the largest eigenvalues, and then within those selected largest eigenvalues components, we select the pettiest components within that set. So technically, here is the if you can see my mouse, here is the final eigenvector we used in this algorithm. This maybe when you're coding is just one step, but it's conceptually there are two steps. And the first step is to condition on this selected eigenvector's direction. We do the projection of the data onto this direction and run the classical PRIM algorithm on it to find the beta model. Beta model is the model with probability beta. Yeah. And then we keep the data within the beta mode as the data points we selected, and gather all these data points from all of the clusters. We get our final reduced data set. And maybe a little abstract, but we will see it in examples soon. So here are some comments for the algorithm. The spirit of the C-prime is that reduce the size of the data in the PTS component direction will return the variations within the principal component at the same time. So they doing these two things at the same time. And the second one is the C-prime, obviously they leverage both clustering techniques and mode hunting to efficiently reduce the large data set. And also you may notice that the C-prime is adapted to various clustering and mode hunting algorithms. You can, it's just you can change to change to other techniques you like for clustering modular or for the mode hunting modular. Yeah. So, and our brain algorithm, our title is spectral analysis, and our algorithm is spectral in twofold. First is the spectral features used for clustering is spectral in nature, and for mode hunting, we also use the, notice here, we use this PTS components here. So it's also spectral. So it's in twofold spectral method. So here we show first the MSTP example is maybe familiar to all of you because it's really a classical data set, handwriting data set commonly used in machine learning society. And we test our method first for label results, seeing that we directly run our algorithm within the use the information from label. So we will see here. Here's the result shown in the TSNE visualization, because don't use PCA here because we already use PCA in our algorithm. We don't want to show some. Maybe I use PCA here. You will thought that there's some relationship between them, but it's not. So I choose the TSNE visualization. It's a information theory based methods, and we will see here the sampling, the color dots here are actually the reduced data set we get in the end. So it's basically doing quite well job, at least from the TSNE plot here. So it's it reduced the size of the data, and at the same time, in general, keep the original distribution, right? So, and on the right-hand side, we select some of the digits and draw these data points out. You'll see from the right-hand side, we see that it do carry very much the variability within each numbers, right? Like one or someone is writing like this in two step. Most people use one step, but with different angles. And some people use bold pens, or some people didn't. And this variability also showing in sevens. There's some seven shape is very weird here. But yeah, and some with a bar here. Yeah, you show plenty of variability within the origin data. And yeah, and then we move on to, if we remove the label and just using the clustering, use the full force of our algorithm without using the labeling information, we'll see the similar thing here. But we will also noticed that there's some misclustering problem happening here because we now using the label. Some place the cluster 71 and some place the cluster 322, but in general it still show the variability in the original data set, and at the same time it reduce the size of the data. Yes, and then we move to our mammography data. This is actually the real data we are interested in. Yeah, the mammography data is mammography is a crucial medical imaging techniques. It is built for early breast disease detection, especially for the breast cancer. For this analysis, we use the mammographic database from the mammographic image analysis society, MIAS, which provides you see here is the sample size is not big. It's only the 322 images from 161 patients, and each image is this size. But actually, I will mention it later. Actually, I didn't use the full force of this resolution, and analyzing the mammography data presents unique challenges due to its high dimensionality and critical need for the accurate diagnosis. Yeah. And for techniques, and we need to add to some comments to it because it's kind of different data, and because I actually run this kind of set of data on my own computer, so I need to deal with the computational problem. So I use convolution to reduce the resolution of the images. So here the full solution is not fully used, and this definitely you can imagine is will have a significant impact on the analysis results, and obviously it's not a positive impact. And also consider the sparse nature of the data, and the ordinary PC is not suitable in this scenario, so we change to the sparse version of Pearson's analysis. Yeah, and the first 10 principal components are used for the analysis, which means in our algorithm we select 10 principal components, and from these 10 we select several PTs one for further analysis. Here is again the TSNE plot for the distribution of the data. You can see it by running our algorithm. We shrink the size of the data, and we still preserve the variability. And you'll see, interestingly here, there's a symmetry there. So for kind of like data island there, we will explain soon what this means. It's actually for blue and the green dots, they are the leftovers, and for all the rest are the red breast. So, and we'll see the plot here. So it's basically the orange is corresponding to here orange. This blue is on the blue one is this one, and the green one is this one, and these two are these two, and this one are this one. You can see there's a pattern here. It's really clear that it's a left and right, and it's kind of a sales matter, and yeah. I'm not an expert in the mammography. I cannot actually, to be honest, tell which what this each single image tell us. Maybe some of them you can see some cells there, and there's cancer cells. I'm not so sure about that, but yeah, but some yeah, some plots you can definitely see some abnormality, right? There's still a variation kept here. Some are more normal, some are not that normal look like. And yeah, and keep that in mind. The next thing we want to, these processes, NLS processes can be applied within some sub data points as well. So for example, here, if we want to look deeper into the green dots in that element, we just focus our attention on group 3 and run our whole algorithm on it, and we will get something like this. Yeah, there's still the same result. The TSNE plot show on the left hand side, and all the color dots show in the right hand side. Actually, here, at least from my knowledge, I can tell the difference between the label 2 and to label 1 and label 3. There's a clear difference that like for label 2 graphs, there is more blurred and most of them are normal. But for 1 and 3, I'm not pretty sure about what happened there, but it seems to me maybe the label label 3 may be more likely to have tumor. But I have a question here, but that's our maybe part of the future plan to collaborate with some expert on this. Yeah, and so the future direction and goals is to continue refining the theoretically justifying the algorithm used. Actually, we have some good intuition about why this is working, but we didn't prove it formally yet. But we'll work on that. I think it's really attractive, and it shows some example that works. It seems to me not bad. And the second one is keep working on the mammography data analysis. Of course, this is not the end of the story here, because and even we get a very meaningful results here, we will still need to, because it's the reduced of the data set, and you need the data reduction is pave the way for the further analysis of the data. So it's just a start of the start, yeah. And to make it more palatable, clinic setting and fully associated breast cancer detection and diagnosis. Yeah, and definitely to make use of the full power of the data, we also need the computational resources from IDSC. But unfortunately, I don't go to that step to borrow that strength. I definitely will. Yeah, basically all the.
Question for Tianhao Liu?
I have a couple of questions. Thank you. A very interesting presentation, and spectral clustering is certainly a really interesting method in terms of identifying similarities between images. And I guess I have a couple of questions, but the first one is the purpose of, I guess, open-ended? What is the purpose of this analysis? Is it just data reduction to reduce data volumes? Because or are you actually trying to assess the breast cancer screening with the mammography data?
Actually, we want to do both, but at this stage we finished the first one basically. Yeah, just finished first one, the data reduction. Because data reduction is not only for the reduction for its own sake, because by these kind of processes, you can make you easier to look deeper into the data set instead of random sampling from it, and you may see no pattern there. So it's kind of like A kind of like more like a visualization techniques used in researchers. You didn't end up there, but it's an important start point. The visual things is more intuitive to human, right? So yeah, to manage the data in a manageable size, and you can simply eyeball it to find something that yeah.
Okay, yeah. Well, one of the challenges with, spectral clustering or other kinds of techniques, unsupervised methods of clustering data is that a lot of times they tend to cluster points together based on large scale similarities. Right?
Yes.
Features. And, I would think, something like breast cancer, right? The cancer may be a very specific, very small signature. And the clusters may actually group together, the large scale features, but may not notice it.
I can
easily
understand. That's the important part for the data analysis. What we are going to do in future is that you must, I think here some pre-processing of the data is necessary. Come augmented for the data to highlight some points or use some other algorithm to leverage their strengths to increase the contrast, kind of like that, and do not have the pretty sure certain answer for that, but we will definitely go into that direction. Yeah.
Yeah, it's very interesting, and I think it would be interesting to contrast, if you were to compare supervised clustering versus an unsupervised method, like the one that you're looking at, whether it would identify similar features for distinguishing, or whether it would actually identify different features that may be smaller, but maybe more discriminating, right? Okay.
Yeah.
You mentioned that you had essentially computational bottlenecks. Can you give us an idea about the size of the problem? Your input images were 1024 by 1000.
Yes, and
is that considered huge?
It's I think it's two party. One is simply because I'm using my personal computer. Second is I'm thinking about the efficiency of kind of computational efficiency of my algorithm, especially for the sparse PCA part. That part may be computational more intensive, and because we are here using the PTS component or like ordinary setting, you simply compute one or two components and you end up there. For PTS component, you must compute more than that, and if they go too far, and because yeah, so that's maybe the part of. I'm just guessing. I didn't have a quite answer for that. Yeah.
Any other questions for Tianhao Liu? Well, all right. Thank you very much. Thank you again for all the participants. Thank you for all the mentors. I didn't see any questions from Zoom. No, I think they mostly logged out. All right. Thank you very much. Thank you for making the fellowship program possible. Thank you very much. Thank you.